Hi, I'm Chris Abel, and this is my project on Agrobacterium tumefaciens. It is a bacteria that can infect plants, and it genetically transforms them to produce food and to provide room and board. It does this by inserting a DNA fragment that integrates into the plant's nucleus, and results in the production of the enzymes and machinery needed to produce growth hormones and food for the bacteria. The growth hormones result in the formation of crown gall tumors, which create more space for the bacteria to live. And it also creates a heavy growth that will fall off the tree and facilitate the spread. That's my conjecture about the ecology of them. So I started with data from the European Nucleotide Archive, as well as NCBI, and I wanted to look at the genomes of these bacteria and compare different strains of bacteria in order to check for candidates that have good potential in biotechnology uses, because the agrobacterium are used to transform plants with custom DNA inserts, which is a very useful technology. So I processed the genomic data using the Bio490 informatics pipeline developed in this class, and how much should I go into talking about the pipeline?
Yeah, you can talk about it. I'm just gonna go through it and see what.
All right. Yeah, so I downloaded the data from European Nucleotide Archive and some other data from NCBI. I checked the completion of the downloads with FastQC. Then I removed low-quality reads with Genomic and assembled with spades. And then I checked the quality of the assembly using Quast by comparing the assembled genomes to reference genomes from CBI. Then I annotated used Proca to annotate the genes, matching them to ontology terms. And then I compared the different strains using Rory and Fandango to generate this figure. The different strains are grouped based on total genomic similarity, and the presence or absence of certain gene all the genes are shown in this figure here. Each blue bar represents a gene, and these blocks have many genes all grouped together, and they are shown from left to right in order of decreasing genomic similarity, of increasing commonality. So the core genome is here, and the pangenome, the genes that are unique to individual strains, are way up here. So after comparing them with that, I also compared some strains that were available on that had genomes already archived at the Kyoto Encyclopedia of Genes and Genomics, and I used Kegg's pathway viewing tools to find these figures. And here I've compared two strains based on the putative presence or absence of several genes involved in virulence. Each of these genes codes for different proteins that are involved in the type 4 excretory system, secretory system, which is over here. It facilitates the export of the tDNA complex into the plant cell, and this strain, the S33, was deficient in many of the genes in that system. So that suggests that it might not be capable of infecting plants, or at least might have decreased virulence compared to the Act5 strain that has most of these genes present, or at least they were matched to known functional orthologs that are known to K. I also used similar methods to compare genes involved in chemotaxis, and they were the same for both strains. So that suggests that they both respond to chemoattractants in the same way. So a priori, I predict that this strain would be more effective at transforming plants, and that both of these strains would respond to chemoattractants in the same way. And I have yet to test that hypothesis.